My name is Virginia Kaklamani. I'm one of the co-directors of the San Antonio Breast Cancer Symposium, and it's my honor to moderate the Year in Review. In the past 4 days, you've heard many talks from many educational sessions and states of the art, and all of these presenters have put a lot of work into these sessions, but I can tell you from past experience that nobody has put more work into the presentations than the 4 speakers today. Trying to get a whole year into 30 minutes is an extremely tough task, and I truly appreciate each one of you for doing this. So to get us started with the basic science and year in review is Neil Vasan from Columbia University, New York.
Great. Thank you so much, Dr. Kaklamani, and also just thank you to you and Dr. Arteaga. This is a phenomenal conference, and it just gets better year after year. So this is my year in review for basic science. These are my disclosures. So for this talk, I chose 10 papers spanning 3 large topics in breast cancer, and really trying to give some new insights into these old questions. Questions around tumorigenesis. Where does breast cancer come from? Why don't we have more breast cancer? Questions into senescence and dormancy, trying to isolate cells that are very hard to see, very hard to measure, with an emphasis on new targets and therapies. And then re-examining, again, pathways that we've known about for a long time, but finding new ways to inhibit these old targets. So 2024 was, I think, a banner year for unraveling the origins of breast cancer tumorigenesis. And I think this mirrors our journey to understand the universe. Each of these studies represents a technological and conceptual leap, going from metaphorically telescopes of old to the Hubble and new cutting-edge ways of glimpsing the universe. And these offer deeper insights into the complex mechanisms that underpin cancer development. So this first study is by the lab of Nick Naveen at MD Anderson, aiming to understand the role of aneuploidy in normal breast cells. So there's been a large amount of literature over the past 5 years trying to understand alterations, genetic alterations in normal tissue types. And in breast cancer, it's been very, very hard to understand the roles of aneuploidy and copy number alterations due to technical limitations. And so to address this question, they applied single-cell DNA sequencing to profile copy number alterations, or CNAs, in epithelial cells from the breast tissues of 49 healthy women. And then they also applied more complicated techniques, including single-cell DNA-seq and ATAC-seq, and spatial transcriptomics. So this figure shows aneuploid normal breast cells in women that are arranged by age. And just starting. Oh, great, thank you. So starting from the top, so most aneuploid cells, this is shown in orange bars, harbored copy number alterations that were clonally expanded, in contrast to the aneuploidy events that were sporadic events, which are shown in light blue. So this next row shows that, very interestingly, 90% of women in this study harbored cells that had aneuploidy featuring loss of chromosome X. So this is a very interesting area of research on how sex chromosome alterations can alter many different phenotypes in cancer, and I think this is a really surprising finding. The third row here, each patient here is arranged by age, and what you can see is that the frequency of aneuploid cells can vary anywhere from 0.3% to 21%. There's a very wide standard deviation, and the median is 3%. And importantly, you can see that aneuploidy in normal cells increases with age. So then they did very deep analyses comparing these copy number events in normal cells with copy number events from TCGA in primary breast tumors. And so what are the similarities and differences here? And this is where things got really quite interesting. They found significant similarities in certain chromosomes, including 1q gain, which encompasses oncogenes and tumor suppressors like MDM4 and CDH1 in ER-positive breast cancer, and then 10q loss, which encompasses the PTEN tumor suppressor in ER-negative breast cancer. So then they used single-cell DNA and a TAC-seq, which combines copy number profiling with chromatin accessibility data to ask the question, what are the cells that are undergoing these events? And what was really surprising is that they found that most of these cells were luminal cells. So there are multiple subtypes of luminal cells. These are the yellow and the blue bars. But you can see that the basal cells, which are in orange, these were not, in healthy breast tissue, these did not exhibit aneuploidy. So where are these luminal aneuploid cells located? So they use spatial transcriptomics, and sure enough, these are located in ducts and lobules. And when they analyzed those cells and performed FISH, those were indeed the cells that were aneuploid. So this study, I think, really challenges our understanding of mutations in breast tissue. It's interesting that some of these copy number alterations are genes we think about in breast cancer, but they didn't identify genes like ERBB2 amplification. So that suggests that there are other copy number alteration events that are necessary for tumorigenesis. And future research will need to explore if patients who have normal breast tissue with aneuploidy are at higher risk of developing invasive breast cancer. I think this study really sets a beautiful groundwork for these future long-term investigations. So then the question arises, if we have all these genetically mutant cells, normal breast cells and of course breast cancer cells, why don't we form more breast tumors? So this is work from the Simons, Shayla, and Van Reenen labs at the NKI in the Netherlands. So this study examines how natural processes in the mammary gland restrain cancer development. So this was a very sophisticated genetic modeling paper where they used confetti mice, and this is a mouse model developed by Hans Klevers' lab. So these mice carry multiple fluorescent reporter genes that are arranged in tandem and flanked by LOXP sites. And so when CRE is introduced, this induces recombination stochastically, which activates these fluorescent reporter genes in individual cells. And this confetti effect is actually heritable. So all the progeny of a marked cell will continue to display that color. So in this way, scientists can do lineage tracing in complex tissues. So what they did here is they injected CRE into the ducts of mice, the breast ducts, and then studied lineage tracing in mammary tissue. And they tracked these clones over time. So they studied this in a mouse that had BRCA1 and TP53 loss, so a breast cancer genetically engineered mouse model. And they studied this both in the cancer cells and in wild type cells. So after about 8 months, tumors developed. But interestingly, most of the mutant cells did not transform and they appeared normal. So you can see here, these are gorgeous images. These cells are mutant, they have the color, but you can see they track exactly in the ducts. They're not forming aberrant structures. And so then when they quantified this in both the mutant cells as well as the normal cells, what's interesting is that both the mutant and the normal clones led to propagation throughout the mammary gland. So this really shows that their model is a bona fide model of field cancerization, field effects of normal tissues. So what restrains this continuous growth? So they found first that short-lived clones with oncogenic mutations die off within months. You can see these curves here. But there is a minority of oncogenic clones. This likely arises from stem cells, and so it supports this idea that short-lived descendant cells can restrain cancerization or can restrain field effects. So secondly, they looked at hormonal effects in the organism. And so of course, this is, we know that the menstrual cycle in humans and the estrous cycle in mice can change breast tumorigenesis. And they showed that when they, using intravital imaging, that when they alter estrogen signaling or alter estrogen levels, they get the appearance of these side ducts, these sort of alveolar structures that are shown in purple. And what's interesting is that these side ducts are geometrically peripheral. They're off the main ducts. They're sort of like small branches of a tree rather than the trunk. And what's interesting is that when they removed estrogen, they removed the ovaries from these mice, all of those cells, that field cancerization effects went down. So estrogen depletion is sufficient to abolish this field cancerization effect. So this paper used a lot of sophisticated mathematical modeling to show that the shape of ducts can shift the growth from a distribution where you would expect the cells to completely overtake the gland. We know obviously that doesn't happen. And when they did this mathematical modeling, they showed that as opposed to this distribution, the log normal distribution where you get these dotted lines, it actually restrains the growth to a more narrow Gaussian distribution. So this is using mathematical modeling to support their hypothesis. So in total, this study used a very sophisticated lineage tracing mouse model to find 3 mechanisms that protect the mammary gland against tumor formation. The first is that most descendant cells, including those with mutations, die quickly. The second is that hormonal changes during the menstrual cycle cause cell proliferation and death. And the third is that the geometry matters. The linear ductal structure limits cell growth. And I think that these findings certainly emphasize the need for identifying early oncogenic clones. I think it'll be really important to validate these studies in humans. We know that the human mouse tissue is very different from the human breast tissue is very different from mouse breast tissue. Estradiol levels are much higher in humans than they are in mice. And so it'll be really interesting to see if we can validate some of these findings in human samples. And so the third really big question here is, how do germline alterations interact with somatic alterations? And so this was investigated in work by Christina Curtis at Stanford, who was one of the AACR award members this year, and she did an amazing presentation earlier this week, and I'll try to recreate some of that. So the overarching hypothesis of this work is that a high burden of germline-derived epitopes in amplified driver genes can deter somatic gene amplification due to the fact that they're presented more to the immune cells, and they increase immune-mediated cell death. So they used a very large number of data sets of tumor and normal sequencing data to examine the relationship of this germline epitope burden and subtype commitment. So they found that individuals with HLA alleles that bind to HER2-derived peptides, these are peptides like GP2, some of these are actually in vaccine trials right now in breast cancer, these individuals were actually less likely, this is shown in red, less likely to develop HER2-positive breast cancer. So if these germline epitopes influence immune editing, then gene amplification should favor the alleles that have weaker MHC class I binding because they would evade the immune response. So they analyzed a group of individuals who had heterozygosity at this locus, at the ERBB2 locus, and they found that the individuals who amplified the reference allele, the normal allele, had a higher amount of differential binding of this epitope. So again, it suggests that these weaker MHC class I presentations can help evade the immune response. So they looked in a large number of data sets across multiple subtypes. Here's shown HER2 positive and ER positive. And tumors, and the patients with tumors with high germline epitope burden, they're more aggressive, they have lower overall survival. When they looked to see at the immune compartments of these tumors, they found that there was an increase in macrophage infiltration and a reduction in lymphocyte infiltration here. And then their overall model suggests that high germline epitope burden might protect against invasive cancer in the precancerous stages, so this is shown here pictorially, but that once the amplification occurs, the cat is out of the bag. Tumors can become more aggressive and develop this immune suppressive phenotype, and that can worsen survival. And this is a really interesting study. It's very provocative, and I think really changes how we think about germline somatic interactions. There's a lot of implications here, I think, in terms of biomarkers and also in vaccine strategies against some of these very peptides to prevent breast cancer. All right, so now I'm gonna shift topics to talking about senescence and other cell phenotypes in breast cancer and in the tumor microenvironment. So we know that residual disease after curative therapy can lead to metastasis, but what is the nature of these cells, these residual tumor cells, and how can we find new targets for therapeutic interception? So before getting into this, I do want to spend a little bit of time talking about some terminology. So we see a lot of terms like quiescence, dormancy, and senescence in the literature, and I just want to say what the definitions that I'm referring to are. So quiescence refers to a reversible state of cell cycle arrest where the cells are metabolically active, but they are not proliferating. And so a subset of these quiescent cells are dormant, and here the cells are viable, but they don't proliferate for extended periods. And these are the cells, of course, that evade detection and treatment by scans or blood tests. On the other hand, senescence is an irreversible state. And it's a permanent cell cycle arrest in response to stress. And it's also associated with the secretion of inflammatory factors that can influence the microenvironment. So it's known that CDK4/6 inhibitors can induce a process called gyroconversion, which is a conversion from this quiescent state to this more permanent senescent state. And it's also known, of course, that the microenvironment plays a lot of roles in these phenotypes, and that cell types called cancer-associated fibroblasts have important roles both in enforcing tumor senescence, but also in becoming senescent themselves. And so this can alter cancer formation and immune evasion. So this first study is from Louis Chodish's lab at the University of Pennsylvania. They examined, using genetically engineered mouse models, they modeled dormant cells after breast cancer therapy. So they use these models, you can see here really beautifully, they get cell kill and then induction of these newer cells, and they characterize these cells. They performed RNA sequencing and found that the top gene signature of upregulation was actually genes in the extracellular matrix. So they took those data and then performed a focused CRISPR-Cas9 in vivo screen, knocking out each of these genes and asking which ones of these regulate the survival of these dormant cells. And they identified this gene called beta-3-gal-T6 as a key regulator of RTC fitness. And so when they, you can see here that these guide RNAs that target this enzyme beta-3-gal-T6 are progressively depleted in the residual lesions compared to the primary tumors. So what is this gene? So beta-3-gal-T6 is an enzyme that adds sugar residues, galactose moieties, to glycosaminoglycans or GAGs. And these are part of proteoglycan assembly in the basement membrane, or excuse me, in the extracellular matrix. And so they found, using a variety of tools, that deleting many of these genes in the pathway impaired RTC survival. So that included this gene, that included some of the downstream genes like heparan sulfate. And interestingly, they characterized this signaling axis linking the outside of cells with the inside of cells through the FGF signaling pathway. And specifically, it involved FGF1, the ligand, and FGFR2, the receptor tyrosine kinase. That's really interesting because they actually pulled out FGFR2 in their screen as well. So I think this is very interesting. It's certainly a really exciting new signaling axis that links what's going on outside of cells with inside of cells to enforce dormancy. And it also, we know that actually FGFR2 inhibitors are now being looked at in breast cancer, in ER-positive breast cancer. And so this suggests that maybe there's an effect not only on the cancer cells, but also on the microenvironment and dormant cells. So the second study is from Sheila Stewart's lab at WashU. So this is a real deep dive into the nature of these cancer-associated fibroblasts, or CAFs. So it's known that human breast cancers have these heterogeneous CAF populations. And so they performed single-cell RNA-seq of 26 breast cancers of all subtypes and identified 3 CAF subtypes, including one called myofibroblastic CAFs, or MyCAFs. And this is a population they did a lot of extensive analysis on. They showed that these MyCAFs have high expression of P16, or CDKN2A, and these P16 positive fibroblasts correlate with recurrence of DCIS that's shown here. So then they characterized these in mouse models. That was in human, this is in mice. They found that there was upregulation of this ligand called NCAM1. And when they looked at the cells that had both NCAM1 and P16, what's really interesting is that when that alteration was found in the stroma, that the cells were completely arrested with low levels of Ki67. But when those same markers are found in the cancer cells, they're not enriched for these senescence markers. So how do these the senescent CAFs drive tumorigenesis? So they found that they do so by inhibiting NK cell function. So they used 2 really elegant mouse models to show this. The first was using a model where they were able to selectively deplete these P16 positive cells and showed that this delayed tumor onset. The second is that they used a senolytic drug, a drug that inhibits senescence. And this is a BCL2 inhibitor. And they found that this was through NK cells. They found that when they actually depleted NK cells, they could completely abrogate this effect. So it shows that this is on target. So then they did a really cool experiment. They actually found the mechanism of how these CAFs inhibit NK cell function. So they found that it's through increased collagen and extracellular matrix deposition. They actually isolated the extracellular matrix that were secreted by these cells and then put them into models where they could study the effects on NK cells. And they found that these senescent CAFs inhibit these NK cells through the extracellular matrix deposition. So overall, I think this is a really interesting study that sheds light on these known cell types in the tumor microenvironment, and certainly with the roles of increasing awareness of senolytics as therapies, it suggests BCL2 inhibition as a possible therapeutic avenue. And so this last study is by Sarat Charnalapati's lab at MSKCC. So this is a new mechanism for gyroconversion to a senescent state. So they performed analysis of patients who were treated with CDK4/6, ER-positive metastatic breast cancer patients treated with CDK4/6 inhibitors. They found that patients with the poorest responses had p53 mutations. When they modeled this in cells, they found that the p53 mutant cells, when treated with the CDK4/6 inhibitor, had long-term outgrowth, but this was not true within the DMSO condition. So then they tried to understand how this actually affects cell cycle reentry. They washed out the drug, and they found that the p53 knockout cells actually resumed proliferation. That's shown here, and this was unlike wild type. They also found decreased expression of senescence markers like HP1 gamma foci in the p53 knockout cells, and this was rescued by overexpression of p53. So again, on-target effects. So what does this loss of p53 really do, and how does it change the cell cycle? So they found that it impairs assembly of the DREAM complex, which is a master regulator of cell cycle gene expression, and this enables CDK4/6 inhibitor resistance. The p53 knockout cells had increased phosphorylation of the subunit p130, and when they overexpressed p21, they got a partial rescue of this phenotype. So therapeutically, what does this mean? So they then looked at combination therapies of CDK2 and CDK4/6 inhibitors. We know that these are also being explored in the clinic right now, and really beautiful phenotypes of long-term growth suppression in these crystal violet assays. They also showed this in organoids and in mouse models. And so then when they looked at patients, what are the consequences of p53 loss? They analyzed patients who had been treated with neoadjuvant CDK4/6 inhibitor. These were patients on the feline trial, and they showed that p53 loss is associated with cell cycle reentry. So I think this is a really exciting observation and mechanistic inquiry into a gene, p53 loss, that could actually maybe select patients who may be better candidates for the combination of CDK2 and CDK4/6 inhibitors. So finally, wanted to talk about the new avenues in the PI3 kinase pathway. We've achieved what I like to term a therapeutic symmetry in targeting this pathway. We have multiple drugs that target all these different nodes. They're FDA approved in combination with anti-estrogen therapies in ER-positive breast cancer, and we have biomarkers of sensitivity and resistance for some, but not all of these drugs. So I think a really important question in the field is why does this only seem to work clinically in ER-positive breast cancer? And one of the reasons for this is for transcriptional and epigenetic mechanisms that result in PI3 kinase inhibition causing increased dependence on ER. So you can imagine it's a seesaw, and when you inhibit PI3 kinase, you increase ER effects, and then you can tamp down on ER and cause synergy. And so one of the mechanisms of this effect is through a histone methyltransferase called KMT2D, which regulates ER-dependent transcription and is actually a direct phosphorylation substrate of AKT. And so the first paper here is from Aneta Tosca's lab at Johns Hopkins, which really sought to identify how KMT2D itself is regulated. And so what they found is a really interesting crosstalk between a methyltransferase called SMID2 and KMT2D. So they found that KMT2D is methylated at SMID2 at this site, and that its phosphorylation, which AKT performs, actually prevents this methylation. And they showed this through a variety of assays in different cell lines. So they then showed that the loss of SMID2 by knockdown or by the loss of its catalytic activity by inhibitors improves the efficacy of many PI3 kinase pathway inhibitors and with and without fulvestrant. And so this is shown both in cells, in organoids, really beautiful phenotypes here, and also in mouse models. And so then when they actually looked at what the consequences are of the methylation itself, when they removed the ability of these or when they knock in a mutation to remove methylation, they found that that increases ER-dependent transcription, again, sort of phenocopying the effects of PI3 kinase inhibitors. And so their overall model shows that SMID2 knockdown and KMT2 demethylation have these overlapping effects, and that maybe this could be a new way of combining a SMID2 inhibitor, which they've tested here, with PI3 kinase inhibitors to increase the efficacy of these drugs. So we've known a lot. So why haven't we been able to move beyond ER-positive breast cancer? There's been a lot of preclinical studies looking at resistance pathways of these drugs, and some of these pathways are shown here. Most of these pathways impinge on two resistance pathways. The first is AKT-independent activation of mTORC1, and the second is overexpression of upstream RTKs. But unfortunately, exploiting these combination therapies in the clinic has not been successful. I think there are many reasons for this, toxicity being one. But moreover, AKT inhibitors have not been shown to be clinically effective in triple-negative breast cancer, where we know that the PI3 kinase pathway is very activated. And so these two studies here really tried to understand what's going on in triple-negative breast cancers and the PI3 kinase pathway. So this first study is from Alex Toker's lab at Beth Israel. So they were really interested in trying to understand if there's synthetically lethal combinations with PI3 kinase pathway inhibitors in triple-negative breast cancer. So they performed a CRISPR knockout screen to identify synthetically lethal genes, and they found multiple genes involved in cholesterol homeostasis, including SREBF1 and 2. These are master regulators of cholesterol biosynthesis. And they used a drug called pitavastatin. This is a statin that's actually very rarely used in the United States, and it's a very potent statin. And they found that when they combined AKT inhibitors with pitavastatin in a variety of models, that this caused cytotoxicity only in ER-negative cells, but not in ER-positive cells. They did a lot of rescue experiments, overexpressing ER, seeing if they could rescue these effects, but it really didn't. And so it suggests that there is something very fundamental about ER-positive breast cancer that is not showing sensitivity to this inhibition. So they did a lot of mechanistic experiments looking at impairing other parts of the cholesterol synthesis pathway, which also sensitized TNBC cells to statins, and these effects were all rescued by lysosomal inhibitors like NPC1 inhibitors. I think this is a really interesting study. It's certainly something that's ripe for clinical development, and I think that there has to be more work done into these metabolic effects in triple-negative versus ER-positive cells. So the second study, again, a very large study to summarize in one slide, was looking at a different epigenetic regulator called EZH2. EZH2 is the catalytic subunit of the PRC complex, or PRC2 complex. This is another H3 lysine methyltransferase. There's actually FDA-approved inhibitors against this target called tazemetostat, which is approved in lymphoma. And they found that AKT inhibitors, when combined with EZH2 inhibitors, synergistically kill triple-negative breast cancer cells and promote tumor regression. They found that when they use this combination, it has effects on differentiation. It enforces the luminal state, and this is dependent on GATA3, which is a master transcription factor in breast tumorigenesis. When they knock out GATA3, they completely abrogate these effects. So there's a lot of biology in this paper. A lot of it impinges on this idea that this combination of this therapy can hijack mechanisms of involution, which is the programmed destruction of luminal cells after lactation. And they identify this JAK1-mediated pathway that impinges also on the STING pathway. So again, a really interesting idea, a really strong mechanistic rationale to combine AKT and EZH2 inhibitors in triple-negative breast cancer. So in the last paper I wanted to discuss was just published in the last couple weeks. We know that metabolic mechanisms can drive resistance to PI3 kinase inhibitors. This is shown here. We know that inhibition of wild-type PI3 kinase by alpelisib and capivasertib can increase hyperglycemia. And we also know that there are many ways to prevent this cycle of feedback, and some of those methods are shown here through diet, through anti-hyperglycemics, through changing dose scheduling, and then more recently through mutant-selective PI3 kinase inhibitors, these two of which have had presentations at this meeting. I think what's really interesting is to think about other ways to have mutant-selective inhibition in other nodes of this pathway, and so this was a paper that was just published by Jack Taunton's group at UCSF. They were really interested in trying to find inhibitors to AKT1 E17K, which is the most common mutation. This, when they did biochemical modeling, they found that the lysine, the E17K lysine, sort of formed this intimate area where where an AKT inhibitor could fit. So they designed molecules to mutant-specific molecules. They showed that the mutant-selective molecules bound to E17K harder and longer than the wild type. And then when they looked in cell lines, these mutant-selective inhibitors caused cytotoxicity in prostate and breast cancer cell lines that expressed AKT mutations, but not in the wild type. Really great work here showing that, of course, that this works also in vivo, but importantly, the AKT mutant selective inhibitor is not associated with hyperglycemia as opposed to the pan inhibitor shown here. And in structural studies, they crystallized this complex. They found a zinc ion that is necessary for binding to all of these lysines in this area. And when they remove that zinc ion by chelation or by a cysteine mutant that can't bind the zincs, they completely abrogate activity. So this is a really end-to-end exploration of a new way to drug AKT mutations. I think will be really interesting to explore in breast cancer and other cancer types with AKT mutations. So this was the year in review. These are the labs who published this amazing work. And of course, apologies to all the other science published this year. And thank you for listening to this talk.
Thank you. Thanks, Dr. Vasan. This was exceptional. So Dr. Pedram Razavi is going to go over the translational part of this year's in review. Thanks, Pedram.
Thank you for the invitation. Really a pleasure to review some of the work that was done on the translational side this year. These are my disclosures. So I would like to start with this slide and thank the organizing committee for paying attention to the translational work. There was much more data presented today in the translational side, and we hope to see more of these type of work in the future. I would like to thank the patients for participating in the studies, both the clinical trials and also translational studies across all of the centers. Our patient advocates who've been very much involved in all the work that we've been doing in the translational side, shaping the studies and making them more relevant to our patients, and also to all the translational researchers who have been trying to narrow the gap between clinic and the bench and find a way back. And I think this type of partnership that we have been seeing more and more in major cancer centers and major centers have resulted in closing this gap even more. So it was a really daunting task going over hundreds of very good papers, many of them published last year. I tried to keep a theme, so I will focus on the liquid biopsy on the first part of the talk, then I will talk on the resistance and response to therapy, and then I have a section just highlighting some of the work on the artificial intelligence. By no means this is an exhaustive review of the fantastic work that has been done last year, and I apologize if there are major papers that are missed from this presentations in advance. So, as you know, liquid biopsy has been transforming the way that we deliver patient care. We can use liquid biopsy in screening for cancer. We are being more and more using it in different types of cancer, and hopefully soon in breast cancer, for detection of molecular residual disease after definitive therapy, and also we've been using it routinely in clinic in the metastatic setting for genotyping cancer and also for monitoring response to treatment. But the major challenge is the very dynamic range in the ctDNA levels, both in the metastatic side and also in early side. The ctDNA levels can go down multiple log in the log scale if a patient receives treatment, effective treatment, and also in early stage, often the amount of ctDNA that you find in a tube of blood is less than the genomic equivalent of one cancer. Often even a fraction of one cancer is seen. So I would like to just highlight this, and you saw some of the other data presented this morning from the ZEST clinical trial. This is the CTRAC-TN trial that was presented. It was a negative study. And Dr. Turner did a fantastic job being a pioneer in this field, trying to monitor the patients with ctDNA testing, but they used the PCR assay, and the assay didn't have enough sensitivity. So they missed actually majority of the patients, and more than 70% of the patients, when they were diagnosed, they already had metastatic disease. And he showed there actually this morning that when you use a better assay, it's still too late. So they used the assay they showed this morning, showed that there is an advantage going to a first-generation ctDNA MRD bespoke assay with multiple genes. You can actually, in this paper, they showed that they can improve the lead time and identify the MRD earlier. The rate of MRD improved significantly in the multi-gene assays compared to multi-mutation assays compared to PCR, but apparently this is still not enough. So there is a significant amount of work now ongoing to improve the sensitivity and limits of detection for these assays. So one emphasis is to increase the number of and the depth and breadth of the sequencing. So, but breadth, we can go, instead of going for 16 mutations, 30 mutations, so on, can go for hundreds or maybe thousands of mutations. As we have more shots on target, the chances of finding them in the tube of blood increases. We can also go deeper and also decrease the amount of background noise that results in decreases the detection rate in this setting. And also another way would be to increase the amount of volume of cell-free DNA that we will test for a patient. So the first work that I would like to highlight is the work that was done by Adam Widman and Dan Landau. And they have previously published a very nice work on the whole bespoke whole genome, not bespoke, tumor-informed whole genome cell-free DNA assay. And they show that they can actually go to the 1 part per million or type of range with that type of assay. In this work, they focused on using machine learning and decreasing the amount of background noise for identifying this SNV single nucleotide variants and also copy number variants in a deep whole genome sequencing of the cell-free DNA. So it's not a tumor-informed, it's a tumor-uninformed assay. And what they showed that they've been able to significantly decrease the amount of noise in the SNV calls. I'm not going to go into the details of what Adam and Dan and the team have done, but they showed that it's mainly based on finding the alterations in the long reads in the normal, and also going back to the cell-free DNA, and also that helped them improve the detection rate for copy number alterations significantly. And then what is significant in the work is that the SNV classifier, despite having no information from the tumor, but decreasing the amount of background noise, it had a good performance similar to what they reported before in the MR Detect assay. So they've been able to decrease the amount of noise by almost 300-fold. And also the copy number classifier, they developed multiple different classifiers for the copy number, also been able to identify the signal from cancer. I think the highlight of the work is when they started looking at triple negative, and this is important for breast cancer. Sometimes even when we do whole genome sequencing of the tumor, we do not have hundreds or thousands of mutations, high confident thousands of mutations, to be able to construct a bespoke assay. He showed here that, they showed here that even the copy number classifier that they developed was able to identify MRD in the patients, patients with triple negative receiving neoadjuvant or adjuvant treatment. So what about screening for breast cancer? This is obviously a hot topic with many assays failing to identify breast cancer early. So this is the work that was done by Dr. Tomasetti and Dr. Vogelstein at Hopkins. They focused on signs or short intersected nuclear elements. These are the non-coding repetitive DNA retrochromosomes, and I'm not going to go deep into the work, but I think it's significant. And they've been able to show that this type of analysis, they found a very unique signature for each cancer, and they've been able to identify multiple cancers using this approach. But still, unfortunately, for breast cancer, the detection rate was extremely low, almost as low as 25%, something that we have seen with the other assays that have been trying to identify and screen for breast cancer using ctDNA. So at this point, we do not have a good cell-free DNA assay to be used for screening for breast cancer but hopefully, with the advances that are happening, we can think about one. Another approach is, this is the work by Dr. Trevor Poole from Princess Margaret. They had a very smart approach going after the p53 mutant germline leaf from many patients. And going to the biology of cell-free DNA, there is a significant amount of information beyond mutations that we can get from the fragment size of the cell-free DNA. We call it fragmentomics in the field. And it's because the cell-free DNA is attached to the nucleosomes. When the chromatin is open and the genes are being expressed, they are not attached. So when the cell dies, the fragment sizes from those sites are small or not existing in the cell-free DNA. They are being chopped by the nucleases. And also, cell-free DNA provides a very good information on the nucleosome occupancy. There are also multiple other features of the fragments that can be used to identify and classify the fragments. And by doing that, they developed a machine learning algorithm to find signal for cancer versus no cancer in this patient, in a normal condition and also in cancer patients. And then they tested the cancer carrier patients. But I think the importance of the work lies in the fact that they went for the high pretest probability patients, for the patients who have high risk of cancer, and by doing that, they improved the negative predictive value and positive predictive, mostly positive predictive value of this type of assays. And I think this highlights the fact that, despite the fact that many of the assays that have been developed for screening, for multi-cancer screening for cell-free DNA have failed to show significance in breast cancer, in general population of breast cancer patients, they could have been potentially useful in the patients who are high risk, the carriers, such as these patients, and have a better performance in this group of patients. And also finding cancer from multiple sites, multiple cancer signal helps in this type of carriers as well. A very interesting work also published this year, going to the even more in the cell-free DNA biology and the fact that the cell-free DNA is being cleared very rapidly by the Kupffer cells and macrophages in the liver, and also by the circulating nucleases. The majority of the cell-free DNA is being cleared by that. So Dr. Victor Adelstensen and the group at MIT, they tried to solve this problem. Honestly, we tackled this problem before I even had studied 2015. I collected samples from plasmapheresis, liters of plasma, to be able to increase the amount of cell-free DNA we're collecting from the patients. But they went for the clearance of the cell-free DNA. And they developed two approaches. I'm going to go over the approaches that they cleared. I'm thinking of this similar to the contrast that we can give to the patients, and he's also presenting it as contrast that we give to the patients who have radiology studies. So we have a CT scan with contrast or without contrast, similar to that. So, this might be a kind of concept that can go there. But what they did, they went to and developed developed nanoparticles, liposomes that are free, that they only have cholesterol inside. They tested multiple of them. And the idea is to exhaust the Kupffer cells so that they cannot pick up the cell-free DNA from the plasma. And then what happened here was that they tested multiple of them, tested the cell viability, and also developed an assay to assess the nucleosome uptake by the macrophages. And they showed that this particular one, the SBE that they developed, increases the amount of cell-free DNA, total amount of cell-free DNA, very significantly within hours, and then after 24 hours, it goes away. In mice, it appeared to be safe, and also they showed that it can increase the amount of cell-free DNA, total cell-free DNA in blood by 60-fold. So just remember, a majority of the cell-free DNA, again, still coming from the normal, and the ctDNA fraction, the amount of the cell-free DNA coming from the tumor remained almost stable. They developed another approach going after the nucleases, and they engineered an Fc silence anti-double strand DNA antibody. And they very also showed that doing this in mice, at least, it was safe, and also significantly improved the uptake, the amount of cell-free DNA in the blood. Again, after hours and 24 hours, this came back to normal. So I can see some utility for this type of approach. There are certain landmarks that we need to have significant amount of cell-free DNA. We need a lot of cell-free DNA from the patients. By having this type of approach and giving a contrast to a patient or the priming of the cell-free DNA, we can increase the total amount of cell-free DNA. It doesn't work for every single assay. We need a type of assay like PCR assays or amplicon-based assays or the assays, capture-based assays that, they go for they perform enrichment for the ctDNA or mutant variants before sequencing. By increasing the amount of total cell-free DNA, we can improve the sensitivity of the assays. So hopefully for the screening, for MRD detection, for identifying subclonal variants in the metastatic setting, this type of approach potentially can help. So in the next part of the talk, I will focus on the resistance and response to therapy. The first study that I would like to highlight is the analysis of samples from Mona Lisa program. I would like to applaud the authors and also Novartis for the commitment for analysis. This is an important effort. They analyzed more than 523 plasma samples from baseline and also end of treatment from the patients who went on Mona Lisa clinical trials, phase 3 clinical trials. And I have one small criticism of the study. I think they could have done a better job in selecting the assay. The assay of the cell-free DNA they used has included more than 500 genes, but the depth of the sequencing and the pattern, the way that the sequencing of the cell-free DNA was done is not consistent with what we use currently in the modern cell-free DNA, ctDNA assays. But nevertheless, this is an important study. They showed significant enrichment for RB1 as expected, and also a span in the tumors in the cell-free DNA end of treatment, cell-free DNA compared to the baseline. They also showed ESR1 enrichment in the post-treatment samples compared to the baseline treatment samples. And they showed very significantly, the changes in the frequency of these alterations in the cell-free DNA as well. But what can we do with the RB1 loss? And I would like to highlight the work done by Dr. Artiaga on the PRMT5 inhibition. So this is one of the dilemmas. So if the tumor completely loses the target and is become RB1 null, how can we overcome this? We should go downstream of RB and downstream of E2F to be able to do this. So one of the ways that Dr. Artiaga and the team did, they did a CRISPR screen to identify in the RB null tumors who were receiving CDK4/6 inhibitor, and the top hit was this gene PRMT5. They showed that PRMT5 knockdown or inhibition results in reversion of the resistance to CDK4/6 inhibitors in these patients. And then, I'm not going to go over the details of this. They showed actually that RMT5 inhibition uncouples the FUS from the RNA polymerase and results in the intron retention of the genes that promote DNA synthesis, and then by doing that, it blocks the G1 to S phase transition. They also showed that there is a synergism between when they combine the PRMT5 inhibition with the anti-estrogen therapies in the cell lines. There is a drug already available for this, and this is a potential way to be able to bypass the resistance that can happen through RB1. I would like to highlight also some part of our work. Neil did a fantastic job going through the lab part. I will go for the clinical genomic analysis that Anton did that resulted in this analysis. I think it's a good example. The whole thing started with the question, who are the patients who would never make it to become an outstanding responder to the CDK4/6 inhibitor? So we started with an outstanding analysis, and we asked these questions. There are patients who are on these drugs for years. Who are the patients who never make it 2 years? And then by doing that analysis, we compared the rapid responses and intermediate responders with this group of patients, and the 2 genes, actually 3 genes, were one of our top hits, so P53, PPM1D, MDM2, and as you know, all of them are part of the P53 pathway. And then we've been able to do a screen, and our screen also shows the same exact thing. And we've been able also to replicate this by analysis of independent, large real-world data, showing that the same exact trend that we are observing here can be identified in the real world data, independent our own data set as well. And Neil presented this perfectly. I'm not going to go over this, but I just want to highlight that this potentially can result in identifying p53 mutation as a biomarker of response to the CDK2, addition of CDK2 inhibitors in CDK4/6 inhibitors in patients. I would like to highlight now one work from Dr. Shamgol in Pedram, looking at the CDK2 inhibitors. A very nice work published in Cancer Discovery. So they report the data on the CDK2 inhibitors versus ATP binding selective CDK2 inhibitors. They show that it results in reversion of the resistance to CDK4/6 inhibitors, abemaciclib, and there was the expression profile, so on and so forth, showed that there was all the reversion was consistent with hallmarks of ETUF targets. Then they showed that what happens when you add the CDK2 is that the cells go back to senescence. So these cells often go to the quiescence, go back to the cycle. These actually cells go, by addition to the CDK2 inhibitors, go to senescence. They did an ATAC-seq that showed that the signature by addition of the CDK2 inhibitors to these resistant cells is very, very similar to the signature in the cells that are CDK4/6 inhibitors sensitive, meaning that the same type of genes are being opened in this setting as well. And they showed that addition of the CDK2 inhibitors in cell lines at the very beginning can actually prevent acquired resistance in long term. One work that I would like to highlight very quickly, and this is going to be a challenge because we're going to have huge amount of data from the large genomic RNA, methylome, so on and so forth, coming for analysis as we are expanding the breadth of the sequencing and profiling that we do in the patients. So this came from Dr. Eidecker and colleagues, and what they did, they looked at the protein assemblies in large data sets, and they asked the questions that what are the genes that potentially result in CDK4/6 palbociclib resistance. They developed a visualized neural network and identified the significant nodes that can predict this. They developed 8 core assemblies to predict this, and then showed that these assemblies are significant both in the patient samples, in the clinical samples, and also in PDXs when they compared to the cell lines. And then they did CRISPR knockout screen, and they showed that majority of the genes that they are associated with response or resistance to CDK4/6 inhibitors are the genes that are identified by the protein analysis, by the neural network analysis, to be associated with resistance. And as you can see, multiple genes here, BRCA2, p53, so on and so forth, are the genes known to be associated with resistance to CDK4/6 inhibitor as the genes here associated with better response. And there is much work going on. I'm not going to go deep into this because we just have a few more minutes. So there is a lot of work ongoing now to try to utilize AI and incorporate deep learning and also large language models into this analysis of this large amount of data that we have in clinic now, and soon we're going to have more in analysis of per patient data to identify and go above and beyond just the single somatic variant analysis that we have been doing, identify interactions and the patterns that can predict outcome in our patients. So the next part of this part of the talk, I'm going to talk about on-target resistance and just highlight 3 papers here. The first paper is by Dr. Todd and Lord. They focused on the on PARP inhibition resistance. They analyzed 47 samples, ctDNA samples from 47 patients with germline BRCA1, 2, and PALB2. And what was different from this analysis from the previous one, they used the modern ctDNA assay that included all the intronic and exonic regions of BRCA1 and 2, and also did a methylome analysis concurrently at the same time. And this work highlighted that the rate of reversion BRCA1 and 2 mutations, these are the mutations that put the BRCA1 and 2 back in frame, actually is much higher than was previously reported. It was 60% in this group of patients. And there were multiple patients that had many reversion mutations consistent with the convergent evolution coming from different sites. But the highlight of work come from this part, that they found that many of these reversion mutations are very large. These are the ones that we often cannot find in their sequencing if we just focus on the exonic regions as we have been doing, or limited exonic regions as we have been doing in the current ctDNA assays. And they showed that 17% of them actually are coming from these type of reversions. They also showed that there were reversion mutations that corrected the splice variants and put it back, corrected by doing that they been able to escape the effect of the splice variants. And also they showed that the methylation, loss of methylation can also do the same thing. So, consistent with work that is going to be the theme in our future translational work and something that adds complexity to the treatment of breast cancer, they also found that despite having a reversion mutation, multiple parallel evolution can result in multiple different mechanisms of resistance, and polyclonal resistance is a significant hurdle even in patients who receive treatments like PARP inhibitors. This paper by Dan Urich and colleagues, actually it's a very interesting paper. It goes for the on-target resistance, and I'm going to highlight two papers in this topic. This is we are talking about now that we have much better inhibitors, much better targeted therapies. Can we see novel alterations, the alterations that we haven't seen before, on-target alterations that result in resistance? So they did an analysis of cell-free DNA and also autopsy samples from the patients who received PI3K alpha inhibitors. I'm showing here one of the autopsy cases, and they showed that some of these patients developed AKT1 loss, AKT1 activating alterations that its downstream results in resistance has been reported before, to result in resistance to PI3K alpha inhibition, and also PTEN loss that has been previously shown also by Dan and us and others that results in resistance to the PI3K alpha inhibition. But it was also this novel PI3K mutations that have not been seen before. And I'm going to highlight the two that they presented. The 859 seemed to be the one that they characterized better and actually results in, sits exactly in the binding side of the orthosteric inhibitors and in the ATP binding site that prevents the cells from binding, prevents the drug from binding to the protein and inhibits the protein. They showed that giving the allosteric inhibitors in this setting can actually reverse this resistance. This is the work that's been done by Dr. Turner and Chalalapati, analyzing the plasma match samples, again finding a mutation in ESR1. This is the F404 mutation. It's a novel, rare variant in ESR1 that resulted in resistance to the fulvestrant through interacting with the binding of the fulvestrant to the ESR1 LBD domain. And they show that 404 mutation by itself actually doesn't activate the pathway. So there is no evolutionary need for the cells to develop these type of mutations before we put a lot of pressure on it. But when we give these cells novel series, it actually doesn't affect the novel series performance. So as we are using more of these drugs, and we're going to find more of these type of alterations, because we're going to analyze the cell-free DNA samples, tumor samples, post-treatment to these targeted therapies, we potentially are going to be identifying this. I would like to, I think these are two good examples that we need to pay attention to such alterations and try to characterize them to see if we can, and identify ways to overcome this. For ADC work, there was not much work published last year. There was some work, but this is one of the very good papers that was published looking at the from the MD Anderson group, looking at the expression of TROP2 and response to Dato-DXd. They showed that the expression of the TROP2 is very significantly associated response in the PDX analysis. These are not tumor patient samples, these are the PDX samples. And then they developed cell lines that they could dose the amount of expression of the TROP2, and they showed that the TROP2 negative and TROP2 positive cells were associated with response to the Dato-DXd. And they showed that adding the PARP inhibitor is something that's being studied actually in mouse, and there is a good amount of biological plausibility for it. Adding to the topo-1 inhibition, adding the PARP inhibition to the topo-1 inhibition can improve the outcomes on the patients who are resistant to the Dato-DXd. In the last part, I would like to go over a few major advancements that has happened in the artificial intelligence, something that we're going to see more and more. And there are multiple papers published in this year on this topic. So one of the major papers that published is the paper that was published by Dr. Fuchs and colleagues from Page. So as you know, at MSK, we have been scanning a lot of our patients. 1.5 million pathology slides have been scanned from more than 100,000 patients. And these type of models are being used to develop what is called foundation models. And these are models that others can use as a pre-trained model. And it's like the human learning mechanism. When we are expert in a topic, learning another topic within that realm of expertise is much easier than someone who's completely naive to the field and has to learn the whole thing. So it's exactly the same thing. So these foundation models have been so far very much limited to the major cancer centers and major companies. But what they did, they developed this model called Virco. There was also one developed by Dr. Mahmoud Faisal and called UNI. But what they did was that, they made this publicly available, and these foundation models, when they become publicly available, they help democratizing using artificial intelligence for the smaller centers, and they can use them as a pre-trained model and develop their own model and hopefully utilize it more and more in healthcare. And as you know, and we have been, is part of our life now, these large language models are becoming more and more integrated in what we do. So this is work by Dr. Mahmoud and team that showed that there are multiple different the different types of large language models. This is the one that they trained on text and also imaging, and they were able to train it so that it can interact with the pathologist, answer the questions, upload the images, and also provide input. Similar to what you would have expected in an interaction with a generalist model like ChatGPT, so on and so forth. And they show that this type of specific models that are designed for the task actually perform better. One major hurdle in translational science and also the real-world data analysis is clinical data. It's the cumbersome task and very manually intensive to annotate clinical data from the charts. So this is the work that was led by Justin G. at MSK and Nikki Schultz. And what they did, they developed the large language model, natural language model to read all the charts, including the notes, pathology reports, radiology reports from more than 24,000 patients. They trained the model, they trained the NLP model based on the Gini data that is very well annotated data based on NLP, and they showed that after the training and applying this to this large group of patients from MSK, the accuracy was more than 90-something% for across the board in different cancer types. And also they've been able to automatically extract a lot of clinical data, accurate clinical data from the charts, visualize it, and allowing translational researchers at MSK to use it. This is actually now publicly available for majority of our cancers at MSK, and we can use it for our research. But one major part of it is that we can do tasks that humanly were impossible. It was impossible for us to read hundreds of thousands of radiology reports, identify the site of metastatic disease, do a time-dependent analysis for the site of metastatic disease development. And this allowed us to do this task and do a time-dependent analysis, identify genomic alterations that are associated with cytoanthropism of disease. So just some quick take-home messages, I think. Higher sensitivity in the liquid biopsy field, higher sensitivity is absolutely needed for detection of MRD in early-stage breast cancer. There is significant ongoing work using tumor-informed and also tumor-naive assays, and preliminary data shows that these assays can perform better than the currently available assays. That novel priming strategy that I showed or similar approaches can help improve the sensitivity of these assays in the clinical setting if they are proven to be safe in humans. For the mechanism of resistance, there is expanding translational research using real-world data and also clinical trial samples. I think it's important to expand on this. It's important to do correlative analysis using right assays in the phase 3 clinical trial data that now we have. We collect cell-free DNA, we collect pre-treatment samples, encourage the patients to provide post-treatment samples, include germline analysis in this type of analysis. I cannot emphasize enough, and that's the whole translational work is based on going from clinic to lab and back, but I think it's important to make this partnership and have this so that our research can become more translational. We need to develop predictive signatures for outcomes, and I think this is a major priority. I described also some of these very rare on-target resistance mechanisms that we might be seeing them more and more. And in artificial intelligence, there's been major advancements. We have now multiple models developed in digital pathology, predicting the outcomes, some of them performing really well. But in the use of these foundation models and democratizing them empowers many of the smaller centers and also us in the major centers when we want to develop the learning based on very small sample sizes to be able to do this, and leveraging AI for the clinical data analysis and all annotations and also analysis of this large data is already transforming translational research and is here for us to stay by incorporating these LLMs into our EMR. So with that, I would like to thank you for the attention and also thank you for inviting me.
Thanks so much, Pedram. So to get us to our clinical part of the update for early breast cancer, Dr. Janice Tsang from the University of Hong Kong is going to give us her 30-minute review.
Thank you, Virginia. Once again, thanks to the organizing committee for the invitation, granting me the opportunity to share the updates on early breast cancer. So these are my disclosures, and because of time constraints, as you all know, I'm a medical oncologist, so I will focus mainly on the medical treatment in early breast cancer in the next 30 minutes. So where we were this time last year for early breast cancer, I think before we left last year from this meeting, we actually were noted about possibility of the escalation of radiation in early breast cancer and also long-term follow-up updates on the main landmark studies, probably the Kettering study as well as the Natalie study. And also the emerging potential of immunotherapy with further consolidation and confidence of the KEYNOTE-522 in triple-negative breast cancer, as well as the KEYNOTE-756 in high-risk hormone-positive, HER2-negative early breast cancer with improved complete pathological response rate, and also larger magnitude being seen for those with high risk being node-positive and high PD-L1 expressed with ER-low tumor. So where are we? 1 year into 024, I would say it's very exciting and a lot of fruitful results. It's actually quite a tough job, as alluded by the other two speakers, that it's quite a challenge, but also a good opportunity for us to review all the data. So is it time for us to out with the old and in with the new, especially towards the end of 2024? I would look at the emerging confidence that we got for immunotherapy, the beginning of optimization of immunotherapy with partnering drugs, and also the emerging role of ACD with IO with further new drug articles and optimization. And this time, are we comfortable enough to remove or take off all anthracyclines? How about to remove all chemotherapy? And also to bridge from what has been heard about the translational research development, I hope that we are engaged and convinced that we have better understanding on biology and potential biomarkers of early breast cancer. And last but not least, I will also explain to you where we've moved from targeted therapy to tailored therapy this time of the year. So 2024 from ESMO is actually very exciting because finally we've got the 5-year overall survival from the KEYNOTE-522 as presented by Peter Schmidt. And actually we have also very good updates and also very nice discussion by Gianpaolo this morning. And as we all know, as you can see, this is actually significant because as you can see, there was a gain of 4.9 actually absolute significance of added value of overall survival. If you look at all the recent data in adjuvant therapy, this is actually top 5 with regard to all the adjuvant therapy. However, it seems that it's only a subset of population is benefiting because even without the advent of pembrolizumab, 81.7% of women could achieve overall survival without the use of IO. And we are talking about added value of this extra roughly 5% of patients. So who are they? And at the same time, we are still having 14.4 women actually being non-responders. So at the moment, we know that Keynote 522 is confirmed that it gives us overall survival benefit. However, how about the most optimization of treatment duration? The value of adjuvant IO, especially pembrolizumab, I think is still not clear, especially is not without immune-related toxicities and financial toxicities. And also the design of the study lacks the second randomization after surgery. And also the optimization of chemotherapy backbone. Is anthracycline a portion? Is this a must? And also, as you noted, the 3-week cycle was used instead of the 2-weekly cycles. And also lack of reliable markers at that point when the data was released with regard to individualized tailored treatment. And could we actually use a bit more the clinical pathological features like TILs to take into account more personalized treatment? How about de-escalation in responders and escalation for non-responders? Or is it prime time for us to replace chemotherapy with combination of ACD and IO? So we are actually very pleased to see the data presented by Joyce O'Shaughnessy at the General Session 1 with regard to exploratory biomarkers from the same Keynote 522 study. And here, as you can see, there is positive association with the T-cell inflammatory gene expression profile with complete pathological response rate and also event-free survival for both treatment arms. And there is actually positive associated with tumor mutation burden with complete pathological response rate and event-free survival in the pembro chemo arm, but actually not the control arm. There's also differential difference that we can see with regard to different molecular subtypes within the triple-negative breast cancers and also the HRD subgroups. This morning, I think we were all here at the General Session 3 with regard to 2 presenters. And firstly is Jeopardoos. Actually, the design of the study is actually very similar, as you can see, basically using atezolizumab with or without, with chemotherapy and compared to placebo with chemotherapy. However, this study seems to be negative, and I will give you some of the reasons and some of the perspectives, and also beautiful discussion by John Paulo. And then it was followed by Professor Xiao Jimen Fudan, Dasha University, that the CAMBRIEF actually study, which is a phase 3 trial, also very similar design showing a positive study. As you can see here, a bit different is there is weekly use of the paclitaxel and also carboplatin with the use of nab-paclitaxel, and also differential added value of complete pathological response gain in the high-risk patients. And as you can see, basically, particularly the Node 3 positive women, they actually got better absolute gain. And there's also tendency of added gain in event-free survival, disease-free survival, and also disease-free survival. This is not the end, but at the end of the beginning. And from what we have learned from the Keynote 522, and now this morning we have 2 extra studies actually looking at the added value or potential added value of IO. So as you can see, the reason why the Jeopardoos was negative, probably because firstly, the standard weekly dose stands chemotherapy was actually allowed for anthracyclines, and also the standard use of capecitabine for the non-pathological response patients, and also the use of olaparib for the germline mutation positive BRCA1 and BRCA2 was allowed. So whether the added benefit was actually going to the chemo or the pembrolizumab or the IO, we do not know the exact picture. And also, what's more, if you look at the CAMBRIEF study, actually, there are some differences in terms of the design. I would say the design is a bit smart, but firstly, it's with regard to even the use of weekly Plaxo and carboplatin. The carboplatin was actually used in the weekly cycle, and also more actually high-risk patients are actually recruited with the T2 above and T3 and N1 to N3 disease. Now, if you look at the result, as you can see, basically for the Jeopardoos, actually the 4-year event-free survival was actually not very impressive, but that was actually achieved by the CAMBRIEF study because, as you can see, the proportion of node-positive women was actually much more, 70%, compared to just about 40% for the Jeopardoos, and just 51% for the Keynote 522. And what's more, it's all the women actually recruited in the CAMBRIEF were actually put on EC dose and therapy. So these are 100% dose and chemotherapy patients, and capecitabine was also allowed for those who are not responsive with complete pathological response after the first part of the treatment. But if you look at the Jipanuvo study that was actually published earlier on, there was some difference with regard to an extra window with the use of weekly durvalumab before the addition of the chemotherapy. And also there is no use of adjuvant IO, yet the result is still positive. So that comes to the point where, is that still a role for adjuvant IO or just a subset of population? I think the more we know how much we do not know. So study design is an issue. How about the lack of biomarkers? And also, it's a heterogeneous population. And how do we define high risk? Even with all the authors from the 4 studies that I've just presented tabulated out, the high risk were actually defined very differently. And there are still a lot of unexplored biomarkers. And also there is a very nice paper published, as I would actually encourage you to look at, from the MPJ Breast Cancer, talking about the added value of dose dense AC versus the 3-weekly conventional, and also the implication when we interpret the result for all these actually IO studies in triple-negative breast cancer. What is more, TILs is a prognostic factor for triple-negative breast cancer, and can we do a bit more and use it with reference in stage 1 disease? And also, there's actually very nice retrospective data from the Netherlands Cancer Registry, as well as actually from a lot of the centers, as you can see, looking at nearly 2,000 triple-negative breast cancer patients who are actually having high levels of TILs, and those with high levels of TILs with 50% more, actually who are not undergoing any new adjuvant or adjuvant chemotherapy, they are actually have better overall survival. So the abundance of TILs is definitely a prognostic factor for patients with early triple-negative breast cancer. It is definitely a potential biomarker. But the technicality issue being the quantification of TILs and whether there's any discrepancy, intraoperative or interoperative discrepancy, and also whether there is added value of AI. What's more, can we spare those with high TILs levels, early triple-negative breast cancer women, from chemotherapy just with IO? So there are actually presentations from the ESMO meeting earlier on this year in Barcelona, looking at new adjuvant nivolumab and also relatlimab, and also from the Bellini's trial. So I would actually encourage you to look at the cohort C and then also the adapted cohort D result. And what's more, I think looking forward, I think we need an international pragmatic patient preference trial like the optimal trial here led by Marlene Koch to evaluate whether adjuvant chemotherapy can be safely omitted in patients with stage 1 triple-negative breast cancer with high TILs. And how about the use of adjuvant IO? And from ASCO this year, we know that the ABRAVE study was actually relatively negative. So 1 year of adjuvant IO seems actually did not improve the disease-free survival for high-risk triple-negative breast cancer. So the high-risk triple-negative breast cancer patients should probably be offered neoadjuvant IO chemotherapy followed by tailor-made adjuvant treatment. And at the moment, there's no data about omitting neoadjuvant IO, but to salvage with IO. So further adaptive design for the study of adjuvant IO, I think we would look forward to the ongoing exploration adjuvant IO monotherapy like the SWOG study. And also, does the order matter? I think at the moment the answer is yes, because the neoadjuvant IO could actually lead to better release of the tumor-specific antigens from the dying tumor cells, leading to better response to the treatment. And also the IO in ER-low triple-negative breast cancer, I think we must actually mention the prominent retrospective study as presented at ESMO meeting this year. And actually this is, despite a retrospective data, in the EU, the pembrolizumab was actually approved, including those triple-negative breast cancer women with the ER-low being between 1% to 9%, and the complete pathological response rate is 75% compared to 65% for the less than 1% definition. However, where should be the threshold for the IHC for all our breast cancer women, and are there any potential biomarkers? With regard to the emerging role of ACD as combined with IO, I think we must not forget the I-SPY2 study that was presented at both the ASCO and the ESMO meeting. So what we have learned is basically we could spare patients actually from extended chemotherapy, and also we could bring them just with IO and also maybe ACD alone or ACD with IO with good response, and then go upfront to surgery and sparing them from further conventional chemotherapy. And the complete pathological response rate with regard to the single usage of datopotamab, the Dato-DXd was presented in ASCO. And as you can see, the de-escalation is basically based on serial MRI as well as the predicted residual cancer burden and core biopsy. And the Dato-DXd is a TROP2-directed ACD selectively that delivers a potent TOP-I inhibitor payload directly to the tumor cells. And therefore, there is potential to have combination with IO for better response of the disease for those actually with high-risk women. What's more, as you can see, the added value of IO to ADC has actually brought an enhanced activity of anti-tumor effect. Now, I think we look forward to probably next year or coming year, the data from Destiny Breast 05 and also Destiny 11, so that we can learn a bit more about the combination of trastuzumab deruxtecan and also its implication of combination of anti-HER2 therapy. And from this meeting, we also have updates on the neoadjuvant HER3-DXd alone and in combination with letrozole for high-risk HR-positive, HER2-negative early breast cancer women from the Zolti Valentine trial. And actually, this is showing us actually women who are put on the HER3-DXd and the letrozole and the tested arm, they have less side effects. So that might be a potential replacement for chemotherapy when we talk about whether it's prime time to actually replace chemotherapy. And what's more, as you can see, there is intrinsic subtype also known for these women. So actually, it showed complete pathological response rate and overall response rate very similar to the standard multi-agent chemotherapy cocktail. And also, there is switch from the PAM50 luminal B to the less proliferative luminal A, or the normal-like at the time of surgery. Now, what's more, as you can see, the safety, as I've mentioned, it seems that patients who are actually randomized to the tested arm as compared to the multi-agent chemotherapy have less side effects, and therefore there's no treatment-related deaths. So the conclusion being treatment with HER3-DXd associated with lower incidence of side effects and also achieving a comparable complete pathological response rate and overall response rate, probably it may be a potential replacement of multi-agent chemotherapy in the days to come. And the Margot study, however, was actually negative. This is a phase 2 study combining magnetuzumab with paclitaxel and pertuzumab. And the conclusion being there's no statistically significant improvement in the complete pathological response rate, but the safety and tolerability were actually similar in both arms. And what is the value of adding a second anti-HER2 drug actually when combined to HER2 ADC? So actually we have, again, Professor Xiao Jimen's team, the Fascinate study, actually looking at the added value of using actually the trastuzumab rezetecan, that's XHR and another ACD, combined with actually chemotherapy and parotinib. And actually the result is showing actually there's a potential to achieve similar complete pathological response rate. Of course, we are still eagerly awaiting for all the upcoming trials as alluded here, the Destiny 05, Compass HER2, and all the other studies. So, it's a prime time. Is it time to say goodbye to anthracyclines in EBC? On top of the SCALA study and the DBCG07 READ trial, I would recommend to read these two papers. This is a very nice, beautiful review by Harold Burstein talking about the added value of anthracycline. And at the moment, I think we don't need anthracycline for the HER2-positive disease women. For triple-negative, I think we still need that, but of course, maybe in subset of patients. And with regard to hormone-positive HER2-negative disease, I think we have also the presentation by Nachan this morning with regard to the impact of anthracycline on those with actually high recurrence score and high genomic profiles, leading to high genomic clinical risk. And also, just to coming back to the story of whether we could actually forgo chemotherapy, Louise and the other Netherlands team also look at 3-year event-free survival of the multi-center phase 2 TRAIN-3 study evaluating image-guided optimization of neoadjuvant chemotherapy in stage 2 and 3 HER2-positive breast cancer. And as you can see, actually, this is again very similar model, and actually the concept is still the same. So the conclusion being 1 in 3 patients with hormone-negative HER2-positive disease and 1 in 6 patients, hormone-positive, HER2-positive breast cancer women could actually achieve complete pathological response rate to only 3 cycles of neoadjuvant chemotherapy. So probably we can optimize the duration of treatment. Probably some women could just benefit as much as the others with standard treatment duration, but with shorter duration and also with excellent 3-year event-free survival and overall survival, and also probably they can go early surgery up front. And there are upcoming studies like the FERGAIN study and Compass HER2, with the FERGAIN also looking at the added value whether patients could actually go up front without chemotherapy in the neoadjuvant setting and also the adjuvant setting. Now, just to updates about what we know, so with regard to the old friends, I think there is still added value of CDK4/6 in the adjuvant setting for the high-risk women. And for ESMO, we have the late breaking from Professor Fashing's team on the updates of NATALY trial on the 4-year overall outcome, which is positive. So NATALY continued to support the benefit of adding 3 years of ribociclib to adjuvant non-steroidal AI in a broad population of patients with intermediate to high-risk HR-positive, HER2-negative early breast cancer patients. Of course, overall survival follow-up is ongoing with a positive trend to be seen in favor of the tested arm. What's more, and also at ESCO, I think Shireen Loy and her team also actually show the prognostic utility of circulating tumor DNA in detection of those women with high risk in the MONARCH-E study. So, of course, we know circulating DNA is prognostic, but whether that's predictive of treatment response, whether that's predictive of response to AI or CDK4/6, that's relatively unknown. But at least we know that for those with baseline positive ctDNA, they do worse, and those switching from negative to positive, they also do worse. So in the context of MONARCH-E, does that mean we need to switch CDK4/6 inhibitor or add back CDK4/6 inhibitor, early switch to third, or with additional targeted therapy, and to optimize with extended endocrine treatment? That we do not know. And as Saf said, we also need the most optimal assay, because at the moment there are different assays testing the ctDNA levels, and whether the detection of early ctDNA levels could be translated to cost-effective modifiable early intervention. I think that is still a question mark, and we need a lot of more data and work to do. And also, as what Ian Kropp this morning has talked about, it might also create unnecessary anxiety if that's positive, and also those with false insurance for those with negative ctDNA. Kevin Kalinske at ESCO also looked at the AMH, the anti-Mullerian hormone, as a potential new biomarkers, and the responder trial. So basically, for those premenopausal women with low AMH, probably they may not actually have the unnecessary toxicities from chemotherapy. So I think we look forward to another upcoming attempt to forgo chemotherapy, as you can see from the offset study, to look at whether we could use ovarian function suppression with AI and CDK4/6 being allowed in comparison with the chemotherapy standard arm. And the more we know, as I've said, the more we know how much we do not know. And the tumor microenvironment is also very challenging and complex. And we have very nice posters earlier on talking about different platforms in clinical practice. So now we have the Oncotype DX, we have HER2-DX for HER2 positive, we have TNBC-DX for TNBC. So I think the tumor microenvironment is very complex and dynamic, and as well as other potential markers should be explored in patient data, especially big data and the real-world data. Immune signatures perhaps are more attractive for integration into the clinic than single immune biomarkers. Of course, that would be the future most likely. And also we need more strategy-based adaptive clinical trials combined with potential valid biomarkers to be designed to tailor-made for all the patient care. Finally, right treatment for right patient at the right time, not just the clinical subtype or the intrinsic subtype, but different population group. So there is also updates with regard to germline BRCA1 and 2 mutation associated with early breast cancer, the Olympia-A study by Judy Garber, and also safety of assisted reproductive technique presented at actually ESMO by Matteo Labertini, and also special groups on ILC in the soft and tech study, older adults with regard to the elderly, the Europa study, the use of exclusive endocrine therapy versus radiation, and also whether we could omit local regional radiation for those with intermediate risk after post-mastectomy, and that's another big group actually study presented by Ian Conkler. So for the Olympia study, this is actually confirmation that there's added value with 10 years follow-up, basically at median follow-up 6.1 years, so we have actually the added value across all the actually primary endpoints. So there's added value of the use of adjuvant olaparib for those with germline BRCA1 and 2 positive. However, for those actually having germline BRCA1 and 2 wild type, there is no added value with regard to new adjuvant use of olaparib from the partner study. There's no added value to combine with Taxol carboplatin, the standard chemotherapy arm. Just to complete the story for this year for the early breast cancer, the underrepresented special groups, so as Saf said, we have the invasive lobular carcinoma, basically with nice presentation by Otto Muska and the team, and also Matteo Lambertini looking at safety of assisted reproductive techniques in young BRCA carriers. That was also presented, looking at the unmet needs for the fertility of this group of women. So, ladies and gentlemen, the end of 2024. Can we out with the old, in with the new now? Indeed, we have good understanding and confidence about IO. This is just the end of the beginning, as alluded by Gianpaolo. Role of adjuvant IO and optimization still need further explanation and further study. There is indeed new plethora of ADCs, and in combination with IO evidence, so further adaptive trials upcoming for tailored treatment for patients is very important. We still need to keep our old friends, our anthracyclines, at least for the triple-negative breast cancer, selected HER2-positive, HER2-negative early breast cancer, perhaps like the impact of anthracycline in high genomic risk node negative disease presented by Chen et al. Earlier on this morning. And we still need to keep our old friends like the AI and also the CDK4/6 in the adjuvant setting, and also the chemotherapy for the time being. We still need our clinical oncologists, radiation oncologists, and surgeons, so we still need the MDT. Indeed, The old concept that breast cancer is a single disease is out, even within a clinical subtype, but now the new concept being each is a rare heterogeneous disease. Even within HER2, triple negative, it's a complete complex disease. And we have now moved from targeted therapy to tailored therapy, meaning from individually with Dennis Lehman's finding identification of the HER2 receptor, and now we are talking about not just precision medicine, not just genomic profiling, but tailoring treatment for a particular patient with a particular biology of the tumor. So from high-C in the past to molecular genomic profile, and also image-guided optimization, and here we see more actually results and or more data looking at zero monitoring with MRI and PET-CT in the neoadjuvant setting. But of course, this is not without cost, because we need more radiologists, we need cost for time, for the manpower, and the cost from the patients out of the pocket. And also we need more predictive as well as prognostic biomarkers. From more is secure to less is more. From escalation to de-escalation, per now, from 2025, we need to tailor-make each of the treatment for each of a single patient, because each patient is unique. The other new trend is AI, as just actually presented by the other speaker. So ongoing unanswered questions, works to be done, a lot to be done. So we still need to come back to San Antonio every year. Optimization of the use of IO to find the specific group benefiting from neoadjuvant and adjuvant setting. Optimization of neoadjuvant chemotherapy, the treatment backbone and duration. When we can be confident enough to forgo anthracycline and perhaps eventually chemotherapy, I think there will be one day, but not now. And further defining tailored management of early breast cancer women from one single common disease to a rare heterogeneous disease. And matching science with affordability is always an issue. Because on Monday, when we go back to our clinic, we'll be going back to each of our countries, different areas. Then we still have problems of how to get early diagnosis of each of the patient, the population, equity of access to health education, diagnosis and treatment, precision medicine with equitable access to MDT care or relevant biomarker testing. So I think communication with our colleagues, our patients, patients advocates, and family members are very important. And thanks to San Antonio meeting, and that's why we have a very nice meeting with the Lancet commissions earlier on the first day of this meeting. And I would encourage all of you just to complete the menu for early breast cancer. Before you go to bed at the end of this year, read the Lancet Breast Cancer Commission. It's a 56-page with a lot of hard work by all our colleagues in this breast community. And with that, I think the way to go forward is, while we are so fascinated about all these different updates and developments, all the new drugs and also personalized medicine, I think we still need to be holistic and come back to a patient-centered approach for aspirational advocacy framework, as alluded from the Lancet Commissions, with different pillars to work with our educations, with the policymakers, and also to be understanding of our colleagues, ourselves, and our patients and families. With that, ladies and gentlemen, so I hope it's been a very fruitful year for early breast cancer, the clinical part, but I hope we could also start active listening after all these news, to start active listening to our patients and the patients' advocates, so that we know their unmet needs, the whole body, mind, spirit, creating a personalized treatment plan. So I don't think we need to drop any friends, but to make new friends and keep the old, one is silver and the other gold. With that, I just want to say thank you to the SABCS Executive Committee, Program Planning Committee. Congratulations and special thanks to all the authors, presenters, and discussants, and salute to all our patients, their families, carers, and advocates, and special nod to all my international colleagues who have been helping me to prepare this talk and also share their insights from different views and from different aspects from a part of the world. Thank you.
Well, Janice, thank you for giving us all a 56-page homework for the holidays, so we appreciate that. And for the last and definitely not least talk for advanced breast cancer, Dr. Michael Danso from Virginia Oncology Associates in Norfolk, Virginia. Thank you.
Thank you very much for the invitation to give this talk. As a practicing oncologist, with a passion in breast cancer research, it really is a privilege to be able to come and give this talk to all of you. So my talk is advanced breast cancer. These are my disclosures. My presentation will primarily focus on practice-changing articles published in 2024. Where appropriate and relevant, I will review data presented at conference in order to give context to the published work. In addition, I will include some limited data on emerging therapies and late-breaking abstracts of special interest. I'll start with ER-positive disease to include HER2-low, then move on to HER2-positive disease, and finally triple-negative breast cancer. We will start with important data in the first-line setting for ER-positive disease to include the Right Choice trial, INNOVA 120, and the SONIA trial. I'll briefly discuss the Abigail trial that was presented at this year's ESMO conference. Second-line discussions will include the EMERALD trial with elacestrant and the postMONARCH trial, and some discussion on the novel SERDs, camizestrant and imlunestrant. For endocrine refractory disease, we will focus on the role of ADCs in the DESTINY-Breast06 data. So in the first-line setting, the addition of CDK4/6 inhibitors to endocrine therapy has been shown to improve and double progression-free survival. This table shows the cross-trial comparison of the 3 approved CDK4 inhibitors in the first-line setting. First-line CDK4/6 inhibitors have also resulted in significant improvement in overall survival across trials with statistical significance with ribociclib and abemaciclib. This table shows the remarkable contribution of CDK4/6 inhibition in improving overall survival, with median overall survival over 5 years in some of these trials. So one of the questions remains for patients with a high burden of disease or in visceral crisis, the question remained as to whether we should be using chemotherapy or this combination of CDK4/6 therapy and endocrine therapy. And this was the subject of the Right Choice trial. Patients with symptoms, visceral metastases, rapid progression of disease, or impending visceral crisis were randomized to endocrine therapy with ribociclib versus combination chemotherapy. Although previously presented, Elsa Herr et al. Published their findings in the JCO this year, demonstrating an improvement in progression-free survival favoring the endocrine therapy ribociclib arm. These graphs show that in patients with or without visceral crisis, endocrine therapy with ribociclib was at least as effective, if not superior, to combination chemotherapy in terms of response rate, progression-free survival, and time to treatment failure. The Abigail trial was presented at ESMO this year. It's not yet published, but this was a randomized patient trial of patients to receive endocrine therapy with abemaciclib versus single-agent paclitaxel in the first-line setting. Patients had to have had at least one aggressive factor listed above here, and the primary objective was 12-week objective response rate. So it met its endpoint in that the 12-week objective response rate was 58% versus 40%, favoring the endocrine CDK4/6 treatment arm. So when we take this together with the Right Choice trial, this establishes that endocrine therapy with CDK4/6 therapy as the appropriate treatment, even in patients with a high burden of disease or visceral crisis. So another trial published was the SONIA trial, and this was published by Professor Sonke and al. Out of the Netherlands and was published in Nature just a month ago. And it challenges the assumption that all patients require immediate initiation of CDK4/6 therapy with endocrine therapy. So SONIA was a phase 3 trial in which patients who had not received previous therapy for advanced ER-positive disease were randomized to receive CDK4/6 in the first or second-line setting. The primary endpoint was defined as time from randomization to disease progression after second-line treatment, otherwise known as PFS2, progression-free survival 2. In this trial, the authors observed no statistically significant benefit from the use of CDK4/6 as a first-line therapy compared with a second-line therapy, with a median PFS2 of 31 versus 26 months, respectively. First-line CDK4/6 inhibitor use was associated with longer use of CDK4 use, so 24 versus 8 months, with obvious implications for total treatment cost. First-line CDK4/6 therapy was also associated with more grade 3 events, and these data challenged the need for first-line use of CDK4 inhibitor therapy in all patients. So how does that work out clinically? So in the real world, in elderly patients with comorbidities and a low burden of disease, it is very reasonable to consider single-agent endocrine therapy as first-line therapy. Moving on to INNOVA 120, this was a trial that was presented and published this year. So INNOVA 120 was also a first-line study, but in PIK3CA mutated metastatic breast cancer with progression during or within 12 months of adjuvant endocrine therapy. It was presented and published this year. Patients were randomized to fulvestrant, palbo, and inavolisib versus fulvestrant and palbo. Efficacy endpoints were progression-free survival, overall survival, and objective response rate. So INNOVA 120 met its primary endpoint with statistically and clinically meaningful improvement in progression-free survival of 15 months versus 7.3 months. Key toxicities in this combination include hyperglycemia, diarrhea, rash, and stomatitis. So this agent is now FDA approved and is available as first-line treatment option for patients with a PIK3CA mutation with endocrine-resistant disease defined as progression of disease while or within 12 months of completing adjuvant endocrine therapy. So, moving on to second line. So, after progression on CDK4/6 inhibition, biomarker testing is strongly recommended, and this doesn't happen enough in our community. Testing can be either through tumor testing or circulating tumor DNA, and we look for PIK3CA, ESR, ESR1, and these other biomarkers. Germline testing is also critical. So in the second-line setting, targeting PIK3CA is an established treatment option, either with endocrine therapy and fulvestrant based on data from SOLAR-1, or fulvestrant and capivasertib based on data from Capitella 291. In SOLAR-1, the median progression-free survival was 11 months with alpelisib fulvestrant versus 5.7 months with placebo fulvestrant. Of note, only 6% of patients in this trial had prior CDK4/6 inhibitor use. Capitella 291 showed a progression-free survival of 7.3 months in the capivasertib fulvestrant arm versus 3.1 months in the placebo arm, particularly in the AKT pathway altered group. So those are two options for patients with a PIK3CA mutation. Moving on to targeting ESR1, in the second-line setting and beyond, targeting the emergence of ESR1 mutations with elacestrant is the standard of care based on the EMERALD trial. Bardia and al. Published an important update in clinical cancer research in October of this year. In addition, results of SERENA-2 investigating the role of the novel SERD camizestrant, primarily in ESR1 mutated metastatic breast cancer, was also published this year in Lancet Oncology by Oliveira and al. And of course, EMBER-3 investigating imlunestrant in ESR1 mutant patients was presented at this conference. As a reminder, the initial results of the EMERALD trial were published in the JCO in 2022 by Bardia and al., where progression-free survival in the intent-to-treat arm population had a hazard ratio of 0.7 favoring the elacestrant arm. However, we see here that there was a 50% early drop-off in terms of progression reported in 2022. We see that very early on, there's a 50% drop-off there. So in an update of the trial published this year, Bardia and al. Describe the patient population that benefit most from elacestrant. So they did a subset analysis, and in this publication, they demonstrated that patients with an ESR1 mutation and prior endocrine therapy use with CDK4/6 treatment for greater than 12 months had a PFS benefit of 8.6 months versus 1.9 months, and that's very favorable compared to the intent-to-treat population, where it was 1.9 months versus 3.8 months. So these graphs illustrate the markedly different progression-free survival benefit according to the duration of prior endocrine therapy CDK4 use in the metastatic setting. This is very practical information for clinicians when selecting appropriate patients for elacestrant use. In my mind, for patients with relatively endocrine-resistant disease defined as progression of disease on CDK4 therapy within 12 months and certainly within 6 months, and who have a significant burden of visceral disease, it would be reasonable to choose chemotherapy or ADC therapy over elacestrant monotherapy use in these patients. The question remains whether the use of the combination of ESR1 therapy with other therapies like ribociclib or abemaciclib, everolimus, or even capivasertib might overcome that resistance pathway. And the results of and there are several trials that are ongoing looking at that, and certainly the results of the EMBER-3 presented a couple of days ago are of significance as well. As I said earlier, there are a number of oral SERDs in different phases of development. These include giredestrant, imlunestrant, and camizestrant. The results of the SERENA-2 study were published in Lancet Oncology this year, randomizing patients progressing on no more than one line of endocrine therapy to camizestrant or fulvestrant. The primary endpoint was progression-free survival, and serial ctDNA analysis were performed. In the subpopulation of patients with detectable ESR1 at baseline, camizestrant produced a clinically meaningful improvement in PFS over fulvestrant. So substituting AI therapy for camizestrant in patients progressing on developing an ESR1 mutation on ctDNA monitoring prior to radiographic progression is the subject of the SERENA-6 trial, which has recently completed accrual. So, moving on to biomarker-negative second-line options. So if there's no ESR1 mutation and no PIK3CA mutation, and you have biomarker-negative disease, options include everolimus-based therapy or switching to CDK4/6 therapy. Everolimus-based therapy significantly improves progression-free survival when compared to endocrine therapy alone, based on the BOLERO-2 trial. And in this year, we had the post-MONARCH looking at CDK4/6 after CDK4/6, and also the EMBER-3 trial, which looked at abemaciclib after progression on prior CDK4/6 therapy. So we will go over some of that data. Now we know that CDK4/6 after CDK4/6 has been looked at in other trials. And the PACE trial looked at palbo after palbo and was a negative trial. The MAINTAIN trial was a randomized phase 2 trial that showed modest improvement in progression-free survival with ribociclib over placebo in patients progressing on prior endocrine therapy and CDK4 inhibition. And again, the majority of these patients had prior palbo. So this brings us to the post-MONARCH trial, which was presented this year at ASCO, but has not yet been published. This was a randomized patient, the post-MONARCH study randomized patients progressing on CDK4/6 and AI therapy to fulvestrant abemaciclib versus fulvestrant and placebo, the primary endpoint being progression-free survival. Progression-free survival was improved from 5.3 months to 6 months with a hazard ratio of 0.73, with a 6-month progression-free survival of 50% versus 37%. Most subgroups benefited, although, again, most of the benefit seems to come from patients with prior palbo, as seen here, but most subgroups benefited modestly. A consistent theme that is again seen is when that it appears to be more of a pronounced benefit in PFS in patients whose duration of prior endocrine therapy was longer than 12 months. Exploratory biomarker analysis showed that there was consistent benefit regardless of ESR1 mutation and regardless of PIK3CA mutation. These are the author's conclusions. Abemaciclib and fulvestrant offers a target therapy option for disease progression on CDK4/6 inhibitor therapy for patients with hormone receptor positive disease, HER2 negative advanced breast cancer, not selected for biomarker status. I think I would agree with that conclusion, although in my mind, it remains unclear if abemaciclib after progression on ribociclib would offer much benefit. This brings me to the incredible presentation from a couple of days ago. This was EMBER-3 presented by Dr. Javeri and all, and this was the EMBER-3 study. Patients were included if they had progressed in first-line AI therapy with or without a cyclin-dependent kinase inhibitor, or if they had recurrence on or within 12 months of completion of AI with or without a cyclin-dependent kinase inhibitor. Patients were randomized to single-agent imlunestrant, standard of care endocrine therapy, or the combination of imlunestrant and abemaciclib. The primary endpoint was investigator-assessed progression-free survival of imlunestrant versus standard endocrine therapy in patients with an ESR1 mutation, or the same in all patients, or looking at imlunestrant plus abemaciclib versus imlunestrant. In patients with an ESR1 mutation, single-agent imlunestrant improved progression-free survival from 3.8 months to 5.8 months, that's 5.5 months with a hazard ratio of 0.52. There was consistent progression-free survival benefit across subgroups in patients with an ESR1 mutation, so whether you had visceral metastases, prior CDK4/6 therapy, or prior PIK3 mutation status, there was benefit if you had an ESR1 mutation. In patients that were not selected for ESR1 mutation biomarker, there was no difference in PFS between imlunestrant and standard of care endocrine therapy. The combination of imlunestrant and abemaciclib, improved progression-free survival from 5.5 months to 9.4 months in all patients with a hazard ratio of 0.57. The PFS benefit of imlunestrant and abemaciclib over single-agent imlunestrant was seen across subgroups, including visceral metastases, prior CDK4 use, and ESR1 or PIK3 mutation status. So there was benefit in all subgroups. There was consistent benefit of imlunestrant and abemaciclib regardless of ESR1 status. There was consistent benefit regardless of prior CDK4 use or prior PI3K pathway mutation. These are the author's conclusion. Imlunestrant single-agent therapy significantly improves progression-free survival over standard of care endocrine therapy in patients with an ESR1 mutation, but that did not reach statistical significance in the overall population. The combination of imlunestrant and abemaciclib significantly improves progression-free survival versus imlunestrant in all patients, regardless of ESR1 status, achieving a 9.4-month progression-free survival with consistent benefit seen across key subgroups. So, what happens once there is endocrine refractory disease? The options of care include chemotherapy, single-agent chemotherapy, trastuzumab deruxtecan, or sacituzumab govitecan. So briefly going over the DESTINY-Breast04 trial, this was a randomized trial of trastuzumab deruxtecan versus physician's choice of chemotherapy in HER2-low metastatic breast cancer who had received 1 to 2 prior lines of chemotherapy, and this was presented a few years ago at ASCO. TDXD significantly improved progression-free survival and overall survival when compared to physician's choice of chemotherapy in patients who had received at least 1 line of prior chemotherapy in the metastatic setting. So, as a reminder, HER2-low is defined as weak to moderate complete membrane staining greater than 10% of tumor cells, but FISH negative. HER2-ultra-low is faint, incomplete staining in less than 10% of tumor cells and makes up an additional 20% of hormone receptor-positive metastatic breast cancer. HER2-zero tumors have no observable membrane staining. So this brings me to DESTINY-Breast06, which was presented at ASCO and published this year as well. So DESTINY-Breast06 was presented at ASCO. DB06 was a randomized trial of TDXD versus physician's choice of chemo in patients with no prior chemotherapy in the metastatic setting. Patients had to have HER2-low or HER2-ultra-low disease. The primary endpoint was progression-free survival in HER2-low disease, with secondary endpoints including PFS in HER2-ultra-low disease. So TDXD demonstrated a statistically significant and clinically meaningful improvement in PFS compared to chemotherapy in HER2-low, improving PFS from 8 months to 13.2 months. PFS improvement was also demonstrated in the HER2-ultra-low population, but there were some adverse events, and the adverse event of note, there were 3 ILD, grade 5 ILD events, so clinicians should continue to be vigilant in monitoring patients for ILD. So this is the author's conclusion, DESTINY-Breast06. established TDXD as an effective new treatment option for patients with hormone receptor positive HER2 low and HER2 ultra low metastatic breast cancer following one line of endocrine therapy. So how do we put this all together? How do I put this all together? So this is an approach to therapy for metastatic hormone receptor positive breast cancer, and this is based on clinical trial data rather than what is available in each of our countries. And obviously, there are some places where a lot of these therapies are not available. But this is how I put it all together. In the first line setting, treatment with CDK4/6 therapy with endocrine therapy is a standard of care based on MONALEESA, MONARCH, and the PALOMA trials. Although the SONIA trial challenges the need for immediate initiation of CDK4/6 in all patients. For endocrine-resistant disease with a PIK3CA mutation, the combination of fulvestrant, palbo, and inavolisib is the standard of care based on INNOVA 120. For second-line treatment, biomarker selection is absolutely critical. Patients with an ESR1 mutation who have previously been on endocrine therapy and CDK therapy for 12 months or longer should be offered elacestrant based on the EMERALD trial. The combination of imlunestrant with or without abemaciclib based on EMBER-3 may become an alternative approach, particularly in patients who have more endocrine-resistant disease. For patients with a PIK3CA mutation, fulvestrant alpelisib based on SOLAR-1 is an option, but my preferred option is fulvestrant capivasertib, as this is better tolerated. For PIK3CA AKT P10 altered patients, fulvestrant capivasertib based on Capitello 291 is the standard of care. For patients with a BRCA mutation, PARP inhibition should be considered. If there's no biomarker present, then the standard of care can be either endocrine therapy with everolimus based on therapy based on Bolero-2, or switching the endocrine therapy and CDK4/6 based on post-MONARCH, and also the recently reported EMBER-3, where the endocrine switch is with the novel SERD imlunestrant. Once endocrine refractory disease occurs, if the disease is HER2 0, then chemotherapy should be offered, followed by sacituzumab, and this is a standard of care based on TROPiCS-2. If the disease is HER2 low or ultra low, one could consider TDXD, followed by chemo based on DESTINY-Breast06. For bone-only disease or a low burden of disease, one could consider Xeloda-based chemo, and then switching to TDXD based on DB04. So that concludes ER-positive disease. I have about 5 minutes to do the rest of it, so we better get going. I'm going to briefly go over the PATINA trial, DESTINY-Breast03, DESTINY-Breast12, and the novel agent zanidatamab in HER2-positive metastatic breast cancer. This is going to be quick. So we know that the landmark Cleopatra trial published in the New England Journal established the role of pertuzumab in the first-line treatment for HER2-positive disease. The addition of pertuzumab to docetaxel and trastuzumab resulted in impressive progression-free survival and overall survival benefit. In the original Cleopatra trial, however, maintenance endocrine therapy was not permitted. But for triple-positive disease, the addition of endocrine therapy to maintenance HP has been shown to improve progression-free survival and time to next chemotherapy, and is routinely done all over the world, including the US. This brings me to the PATINA trial, and this was presented yesterday. This trial randomized patients with triple-positive disease completing induction THP to HP endocrine therapy versus HP endocrine therapy with palbociclib. The dose of palbociclib in this trial was 125 milligrams, 3 weeks on and 1 off. Results were striking, with a 15-month improvement in median progression-free survival from 29 months in the control group to 44 months in the palbo group. Overall survival data is not mature yet. The author's conclusions are above. Palbo added to anti-HER2 and endocrine therapy may represent a new standard of care for patients diagnosed with hormone receptor-positive, HER2-positive advanced breast cancer. Taken together with the MONARCH trial, these results establish the addition of CDK4/6 inhibitor therapy to maintenance HP and endocrine therapy as a very reasonable new standard. So in practice, some of us are doing this already. So I do look forward to seeing the peer-reviewed publication, however, and I am particularly interested to see if efficacy is maintained with dose reduction of the palbo. A lot of us in the real world use a much lower dose of palbo at 75, and then this trial was done at 125. It'd be interesting to see whether the efficacy is maintained with a lower dose of palbo. Going to move on briefly to the Destiny Breast 03 trial, and this was a second-line study, and patients were randomized if they were progressing on first-line therapy to TDXD versus TDM1. This trial was originally published in the New England Journal of Medicine, but an important update was published this year by Cortez et al. And these are the findings at a 41-month follow-up: significant improvement in objective response rate, significant improvement in progression-free survival from 29 months compared to 7.2 months. Survival was 52 months at TDXD versus 42 months with TDM1. Similar grades of toxicities, and about a 16% rate of ILD events with no grade 4 or 5 ILD events. Briefly, going to talk about Destiny Breast 12. This was another important trial presented and published in the Annals of Oncology this year. And this was a phase 3b/4 single-arm, two-cohort open-label study of TDXD in previously treated HER2-positive metastatic breast cancer with or without brain metastases. Subjects received less than 2 prior lines of therapy in the metastatic setting and were tucatinib naive. The primary endpoint was progression-free survival, with additional endpoints including objective response rate and overall survival. So these are the results of this published data. 12-month progression-free survival was 62% in patients with stable brain mets and 59% with patients with active brain mets, with median progression-free survival of 17.3 months. In terms of objective response rate in patients with no brain mets, the objective response rate was 62%. And in patients with baseline brain mets, the CNS objective response rate was 71% for all patients, but 79% for patients with stable brain mets and 62% in patients with active brain mets. TDXD showed consistent 12-month overall survival with 12-month overall survival of 90% in patients both with or without brain metastases. So how do I put this together for HER2-positive disease? Well, first-line therapy is clearly THP still. For triple-positive disease, maintenance therapy with endocrine therapy and CDK4/6 significantly improves progression-free survival and will become a new standard. In the second-line setting, patients with no known brain metastases or stable brain metastases should be treated with TDXD based on the results of DB03 and DB12. On progression, they should go on to receive tucatinib-based therapy. In patients with active brain mets, treatment options include tucatinib, capecitabine, and trastuzumab, but with the new data from DB12, it is very reasonable to consider TDXD in this setting. In the interest of time, I'll skip this novel agent, and I think I have about a minute to do triple-negative breast cancer. So we'll briefly talk about the ASCENT trial, DB04 subgroup analysis, but in the interest of time, I'll skip DB04 because we all know that data. We know that it improved, we'll skip, the ASCENT trial because we know that data. We'll talk briefly about the DB04 subgroup analysis. So in the subgroup analysis of DB04, we saw that the use of TDXD was in a small subgroup of patients, 60 patients who were HER2 low and triple negative. It was a small group subset analysis where the median progression-free survival was improved with the TDXD. But again, it was a small subset analysis. Nonetheless, this is clinically meaningful and has been accepted by some to justify the use of TDXD as an alternative to sacituzumab in patients with HER2-low triple-negative breast cancer. But again, this is a small subset analysis and should be taken with caution. There are a bunch of new novel agents, datopotamab deruxtecan and darolutamab in advanced triple-negative breast cancer. The Begonia trial showed a remarkable response rate of 73% in patients with PD-L1 positive disease or PD-L1 negative disease. There are a bunch of new bispecific antibodies targeting both PD-L1 and VEGF, and these show remarkable response rates. They're not quite ready for prime time, but certainly in the upcoming meetings, we will see more of these agents. Key ongoing phase 3 trials of TROP2 ADCs in advanced breast cancer include TROPION-Breast02 and ASCENT-03 in IO-ineligible patients. And the combination of these agents with IO is the subject of the ASCENT-04 and the TROPION-Breast05 trials. In the interest of time, I will not go over this, but basically for PD-L1 positive disease, chemotherapy pembrolizumab is a standard of care. If they're PD-L1 negative, chemotherapy or olaparib if they have a BRCA mutation. And in the second line setting, sacituzumab is an option, is the standard of care, and TDXD is an option if patients have HER2 low disease. In the third line setting, we could see the list of agents we could use in the third line setting. My final slide shows the ADCs in development. The top line shows the target. The second line shows the monoclonal antibody, the payload, the linker, and the drug-to-antibody ratio. These are all in development and are very promising in triple-negative disease. Finally, I'd just like to thank a bunch of people at Virginia Oncology Associates, these physicians and clinical researchers, including Angel Kidd, who helped me with these slides, Dr. Nina Balanchivazzi, who helped me go over some of the important data. At SCRI, I'd like to thank Joyce O'Shaughnessy, Erica Hamilton, Mabel Merdonis, Denise Yardley, and Jay Anderson, and Maithili Shastri, who does an amazing job for us at SCRI. I'd like to thank my mentors, Andy Seidman and Cliff Huddis at Sloan Kettering, without who I would not be a breast cancer oncologist. Special thanks to Seth Wander, who helps me with trying to understand ER-positive disease better, and the nomination committee, including Dr. Pat. And finally, a close family member, Clement Knight, who is the reason I'm an oncologist and is no longer with us. So thank you for your time. Thank you.